think we're live. All right. Hey everyone. So welcome to our user spotlight webinar. Just wanted to quickly say, we appreciate everyone for registering and attending the webinar. For the next 20 to 25 minutes, our head of biology at Big Omics, Axel Martinelli, and principal scientist Ken Steadman from Plexium will be sharing their insights and experience with omics data analysis in cancer drug discovery. They'll be going over how they've approached omics data analysis during their time in research and some of the key challenges and successes they've experienced when it comes to creating infrastructure to address these analyses. Just quickly, if anyone in the audience has any questions or comments, feel free to drop them in the chat below, and we'll take a few minutes at the end to address them. And without further ado, I'll leave it to our speakers to take the floor. So go for it, guys.
Okay, thank you, Griffin. And it's really nice to have you here today, Ken. So
nice to be here.
We have known each other for a while now, and I've always been actually curious a bit about your academic background. How did you get started in the world? Well, first of all, of biology in general, and then omics. I think you hinted something about that. It would be nice to hear it again.
Yeah, I got started actually working at the National Cancer Institute, and we had a lab that was really spanned both. On one side of the lab, we were doing discovery. We were looking at the SEER database correlated with expression profiles in SAGE to come up with new targets. At the same time, we were actually doing we were doing phase 1 and phase 2 clinical trials on epigenomic modifiers. So we kind of covered the full drug discovery thing, including discovering one of the major multi-drug resistance transporters. So it was like this whole spectrum from coming up with new targets to making better, overcoming drug resistance to actually in the clinic getting a drug approved. So that was like such a great, primer of like the whole spectrum of cancer, drug development. And then from there, I went to Georgetown, where we were a heavy clinical lab. And so there we were looking at ways to combine drugs we had, and then a lot of clinical diagnostics, and which was ended up, big data there. We're, 3,000 patient, looking at, SNPs analysis and how they would relate back to drug resistance. And then I finally did some basic science research at Cedars-Sinai. So I've done every point of drug development, from coming up with a new idea to new targets to, hit finding to drug approval, just never in the right order, like all over the place. But it's nice because you can kind of stitch together, like, oh, in the clinical side, we do this, and kind of prioritize those new target development. So it's kind of been an interesting career, hopping all over the place, but it's all been drug development. It's all been cancer.
That's interesting actually to hear that, because my background was also on drug resistance in malaria. So we have something in common there for sure. And my understanding is that you really started as more like a molecular biologist, right? Not as a bioinformatician.
Yeah, I think I really started off as a pure molecular biologist. The bioinformatics came from new target generation, and then a lot of just hypothesis testing. You can read in a paper, but at the end of the day, there's gene expression omnibus has so much data. There's so much data out there. If you have just kind of a simple question like, hey, if X happens, then Y follows Z, you can usually repurpose data to follow that Y and be like, okay, well, if that was true, and if I find a data set of this, this kind of tumors, we should see this, happen. And so then just use, so you end up just kind of answering your own questions using data. And then you realize so much has been done, and it's just using data, orthogonally in a way it wasn't designed, slicing it up to address your questions. And, you can get a pretty good idea of whether or not something's going to be valuable or not using, repurposing data. And so really, and then just during my PhD, it became a thing where it was just, I wanted to kind of do my own bioinformatics, primarily just because it's quicker and easier, and you can do it from, I'm not dependent on somebody else. So then, your network analysis and that kind of stuff kind of came along. So I'm still a biologist, but I'm a biologist that understands bioinformatics, can do it a lot slower. Setting up a pipeline, it's probably been 6 years since I set one up. I could do it, but obviously someone, there's a subject matter expert that's going to do it faster and know it better. But knowing how it all kind of comes together can help you design, the project to get the best answer.
Indeed. And it was interesting before you told me how you suddenly were introduced to the world of omics data analysis, and back then it wasn't really known as omics data, right?
Yeah, I don't know if we called it informatics or big data, but even then, SEER, had survival rates for all the tumors for years and years, huge amount of data. And we came across SAGE, and we said, what if we can link survival to expression profiles, make a fingerprint, of something. And we called it Wilfred Stein, who actually does drug resistance in malaria, which I think is a funny, nice.
Yes, indeed. It's quite coincidence.
Came up with he called it the axis of intractability. And so then we just hunted down, the edges of that axis, like what's easy to treat and what's hard to treat and what makes them different, to kind of come up with new targets.
Oh, that's interesting. And now, of course, you work in the biotech world, and I think you deal a lot with various types of omics and data, especially transcriptomics data. So in your line of work, now that you work for a biotech company, obviously the priority changes, that's you put more emphasis, say, on speed rather than, how thorough you can be like in the academic world. So what sort of challenges did you encounter when you first entered the biotech world when it comes to transcriptomics data analysis?
I mean, speed is, key. Where I work, Plexium is a small company. The way we beat big pharma is agility. We've got to have our decision loop, much, much tighter so that we can generate and move forward. We're competing against, 10,000 people companies. So you've got to have, be agile, and you're going to make decisions quickly. And, that's kind of what I didn't like about academia, which, it's a double-edged sword, is you do have that time, but also you might tackle some things that are really tedious just because labor is cheap, and, you're not going to be scooped once you're out on the fringe of, data. So here, labor is an important thing. You can generate, huge experiments, but is that really worthwhile? So it's really about focusing, designing a project and experiments to give you that best answer. But also, since, you're working, proteomics and transcriptomics, binding assays, and, cut and run, all, integrating them, you've got to, kind of design that. So, your kinetics for transcriptomics are going to be totally different than proteomics. So you've got to kind of make, some, compromises and evaluate, like what's going to really give me the best, data so that we can make that good decision, rapidly. So, it's always a compromise. And, the other thing is, the integration science, I think of it, I like to think of it as it's the teamiest of team science, right? Because no one man is an island. You've got the proteomics experts, the transcriptomics, your RNA core, the proteomics core, all integrating together. And so you all kind of have to, work together, pick your compromises and to generate that data quickly. And then, the other problem is you don't want siloed data. That's the other challenge.
Yes, indeed. And I definitely want to go on to that aspect. But quickly, a question that came to my mind now that we are talking about academia versus biotech sector. Just a little fun question. But one thing that you miss about academia and one thing that you really like about the biotech life?
I don't miss much from academia. I mean, honestly, I loved government lab. I thought NCI was, really great. I guess there's kind of academic time. I think the government labs are the best, compromise, because you're funded, you're not begging for money and grants, but you still have big team science. What I like most about academia is, I mean, a lot of people say, oh, there's, total freedom in academia, but you still have to pick stuff that's going to get granted. You can't just, pick something totally. I think academia, I kind of enjoy, or in industry, I kind of enjoy that, where you kind of come up to a thing and you're like, oh, that's an interesting problem, but can I triage that into something, And so it really helps you focus, focus on experimentation, focus on, project planning, on like, how can I design something that addresses that without wasting, some PhD or undergrad, 8 months of them generating. Like, how can I address this? Get 80% of the question answered in 2 weeks instead of making this some long drawn out project for 4 months. So it's a challenge, but it's also, you realize you can be a lot more agile.
Yes, I think this is a really great answer. So going back to what you were saying before about the siloing of data, the importance of integrating data, but also, the challenges that you are currently facing when analyzing omics data, especially in a biotech environment. Can you tell us a bit about that?
I mean, there is data tends to be siloed. You design data to address, focus on a question. You can't have a massive generalizable experiment, because it's just, they take a lot of time. You go into an omics black hole where you just sifting and sifting and sifting. So you have to be focused. But then there is kind of the problem where you design the experiment, you get that one little answer, and you put it away, and you never touch it again. And that's not really extracting, the value, the labor time, the costs out of it. So I think what's, for me, it's about like not having that siloed data. So generating data that I can go back to, drug development, it's an evolution of compounds, it's an evolution of targets. And so being able to rapidly go back to old data and compare it to new data. So you can say, oh, okay, now we've cleaned up this, or, oh, we're seeing this new thing, or, oh, this is cleaner now. That's really important. And the other thing is being able to access the data quickly, because you might go to a talk, you might go to a paper, and with my old system, my own homegrown data set, I would really have to make those decisions like, how important is this? This is a half a day of work to go back in there. I've got to pull up old code, got to like shake the cobwebs out of my brain on what that code was. And so, that ability to kind of go back to data real quickly, oh, I went to this, oh, I read this interesting paper. Are we constantly seeing that effect? Loading in the data, opening up, addressing that question in 15 minutes and closing it and being done, and, putting it out in your notebook, make a little slide. That's really important to extract that value out of that experiment, rather than just like, I addressed one question and I'm done, I'll never look at it again. The ability to go back and, really have that evolution of data.
That's actually a very nice point that I think it's valid both for the biotech and also the academic sector. And on this point, at some point during your work, did you think, oh, maybe it would be nice to have a third-party solution, like a platform that could really help us speed up our recovery of the data and our reanalysis of the data based on old and new data sets? So at some point, did you start thinking, yeah, we could do with a solution like that?
Well, we were kind of at a nexus. We were generating a lot of data, and we hadn't really decided. We were like, well, how are we gonna do this? We had some kind of homegrown systems, but we recognized that those homegrown systems had already been outgrown. They were, you couldn't, it's not just generating the data, but it's extracting the figure. That last mile is really important. You got to simplify the data to address that answer, for it to go out for other people to understand, like being the king of the data, on your single island doesn't really help, anybody. So our kind of homebrew solution wasn't working for that last mile. We didn't, and then we just, it's kind of kismet. We ran into Big Omics, and then, we kind of started working, and it's kind of been this process where, as we both, I feel like both of the companies have kind of grown as I started doing the kind of harder and harder problems. Big Omics, the platform got, more and more in depth and focused, on what I needed to get out of it. So in some ways, it felt like Big Omics has been my custom solution, like, it's because it's like, I need this now. What about this? And it's like, oh, hey, and then put in a ticket, or discuss, make a slide deck, and so it's kind of grown to fit where I need it to fit. So it felt like a very custom solution.
Oh, that's great to hear. So obviously, I gathered from your word that the platform did help you address some of the issues that we discussed and some of the challenges that you were facing with your data analysis. So what would you say was the impact before and after the platform?
The biggest thing is that speed, like being able to go back to data, you can answer a question in 7 minutes, and that's just, I don't know, I'm not organized enough, or my code isn't good enough, that I just, I don't have my triage for how I'm going to handle a project, how I'm going to handle a question is much easier now, because I can just go back and quickly look at something. And it's nice to be able to easily compare a data set to a data set. That's it's a trivial thing. Being able to compare how this drug did in this instance versus a different drug that was related in a slightly different instance is a trivial experience. Otherwise, I can't imagine, that's a full day process, like making sure, that, you have to even approach, okay, how am I going to write this code? Whereas that I can just do that in, like I said, 7 minutes, and it's a trivial thing. I get a figure, and it's done, and that's it. Boom, you're done. Figure, slide deck, it's over. And so that's a real, time saver.
It's really great to hear, and I already know what kind of are your favorite modules and functions on the platform. But if you could share again with us, what sort of features you really like the most about the platform.
The best one is the biomarkers, because it's like it's just cheating. It's like 4 clicks and you're there. It feels wrong. It's so simple. Because it's like, I even, said something to our VP. I was like, I don't know, sometimes it just feels wrong. Like, you're paying me, because it really is, getting a biomarkers is trivial at this point. You structure the data the right way, pull into, load it in, set it up, get your contrast right, and boom, your biomarkers are there, and then you can go on. That's a 15-minute max, proposition. So that's a huge time saver. Normally there would be like 3 days, and then going back and carving and making sure and rechecking. And so now it's, you can have the biomarkers in 7 minutes, and then another 7 minutes to kind of confirm and look at what else you're getting and run it back through some old data. But like I said, 15 minutes, that's a huge time saver.
Yeah, no, it's absolutely, that's definitely, as you mentioned, one of the main aspects when you do research in the biotech sector. So out of curiosity, because I remember you told me last time how you like to interact with a new data sets when you put it into the platform, and it's kind of different for how I do it. So can you just explain, like, once you have a data set, what are you going to explore first? What sort of going to drive your exploration of the data?
At this point right now, it's boom, biomarkers go in, and then I want to see the structure of the data. Did we have because from a pure bioinformatics, you're looking for p-values and quantitative, and a biologist, you're looking for qualitative pathways, and it's almost like themes. You're kind of looking for themes that pop up, and so the ability to kind of look at that data from kind of holistic thing. Like obviously the biomarkers are important, but I like to kind of jump back and forth, and I might have a handful of genes. I'm going to go right into the the data set and look at those genes, and right off the bat, see how much variation we have on them, to kind of qualitatively compare this to a previous experiment. You're like, oh, did I hit these targets? Are they going down really well? And then I'll go back and then do, a Spearman correlation off of those and see, oh, okay, is there anything new that showed up in this theme that I'm kind of looking for? So it really is like, kind of almost stream of consciousness. You're hopping from module to module to module. And then as something comes up, oh, that's interesting. You might kind of rabbit hole on that data back. You might, take some notes and then go back to that. The other thing I really like is not just taking screen captures, but opening, open as picture, and then I'll have them like ordered across my tabs so that I can kind of go back and order them together and be like, here's how I'm going to put this together. Here's how this story goes together, across my tabs. I think that's a really interesting, which, in R, I would never do, that's just like, you couldn't possibly put together a storyline of tabs, you know what I mean? And with actual figures in them, R is an iterative loop. It's I'm doing one thing, stop, come back to a new, question, that I need to go through. So it's a totally different, idea. And it's a little bit more freeform. You might probably catch something new because you're not focused on making sure the next line of code works and, syntax or something like that.
Yeah, I know the frustration you're talking about. And I actually like what you say, that it's almost like a stream of consciousness analysis of the data, which, it's kind of freeing, especially as a biologist, where you don't have to be, sort of limited, like, okay, can I translate what I want to find in a code line, and can I implement it without having to rewrite it 10 times because I forgot a comma or something? And, it kind of mirrors my experience working with the platform as well, where you sort of sometimes let the platform lead you, as you say, down a rabbit hole of things that you weren't expecting to find. So have you actually had this kind of experiences when working with new data, where suddenly you thought you were looking at a certain area or a certain aspect, and suddenly the platform opened up a sort of new kind of worms, so to speak?
Yeah, I mean, it's pretty common, and you kind of collect those because, since you're kind of working on the same target in the same chemical space, you do develop these kind of themes, like, hey, we saw this in this data set, and that even goes back, to apply forward, like, okay, we got to keep an eye on this in an animal model, So there's a definite serendipity that you can get out of looking at data this way that I can't get out of plowing through R, because there really isn't such thing as serendipity in R. You might have a thing, but that's gonna be a new piece of code that you've got to write. That's got a new module I needed to load. So there isn't that chance encounter.
Yeah, no, indeed. It would be more like AI applied to R that would maybe allow you to do that.
Yeah. Well, I hate to interrupt the discussion here. We're running out of time, so just want to make sure we leave.
I can't. Is Griffin?
Yeah.
Can you not hear me?
I don't hear Griffin.
Okay.
He's speaking now.
Actually, you can relay. You can relay the question.
I'll relay the words.
Just want to make sure we left a couple minutes for questions. Doesn't look like there was too many audience questions, so I have a question for the both of you here. What developments are you most excited about in biotech going into 2024?
Okay, so there is a question for you, Ken, from Griffin, and he's asking, what developments in the biotech world are you most looking forward to for 2024?
I mean, everybody's hoping AI is going to solve everything. It'll be interesting. I do think certain fields are better adapted to others. I mean, I feel biomarkers are something that's really, AI is, really primed to help. You can take different, totally different type of data, and it can, AI will be able to draw inferences that even the most clever, rat catcher could never come up with. Like you just, but it also, it's going to be kind of tricky. Like it's overuse in other types of field might generate, some kind of, garbage data.
Yes.
I just read within the last like 48 hours a paper that was talking about AI is already reading AI, so we've already kind of come full circle where ChatGPT is reading stuff that was generated by ChatGPT. So you can see where there's a real opportunity for garbage to be created too. So
and propagated, absolutely.
Yeah, and then obviously in the TPD field, now we're really kind of waiting for the the first kind of tranche of patient data to come back, and I think that's going to be really exciting. It's a brand new field, and there's still some big questions that need to be addressed, and so for my purposes, I think that's really exciting.
Awesome. No, and I agree with you. I think there is certainly a lot of promise there in AI. As you say, biomarkers discovery, hopefully also for drug development. Let's see if you can live up to the promise. I hope so. And certainly we at Big Omics are keeping a close eyes on that. And you might see some AI biomarker discoveries in the future. I'll certainly make a point of that.
A new tab, I would love it.
I know.
We can get my 7 minutes down to 4 minutes or 3 minutes.
Absolutely.
Right. Well,
Griffin, you want to wrap up?
Yeah, I'll wrap it up. I don't think Ken can hear me right now, so just you can relay this afterwards. But thanks again, Ken, for joining us and discussing the trials and tribulations of data analysis. We're grateful for all your insights, and we wish you the best of luck on your journey through the ever-changing biotech landscape. And just a quick wrap up. As Big Omics, we support biotech and pharma teams in omics data analysis by providing highly interactive and user-friendly analytics for transcriptomics and proteomics data. So if you'd like to learn a little bit more about the Omics Playground platform, you can visit our website at bigomics.ch or reach out, and we'd be happy to walk you through the platform and its functionalities. So thanks again for everyone joining, and make sure to follow us here on LinkedIn to not miss out on our next webinars. All right, have a good one.
Okay, so just Griffin was wrapping up, and he wanted to thank you personally for participating, and of course the thanks come from not just him but from all of us for giving us your time, and then it was quite an interesting chat that we had. So I personally certainly enjoyed it.
Yeah, I was actually. It was interesting to hear that you were doing malaria drug resistance. And
yeah. We definitely should have a chat about that at some point in the future. Yeah. It's definitely a lot of there is a lot of correlation between how we were studying malaria drug resistance and drug resistance in cancer. A lot of things were like you didn't only take ideas from malaria; we also took a lot of ideas from cancer research.
That's interesting. We'll have to compare collaborators.
Compare notes, absolutely.
All right, I think we're all set. It was a solid webinar. Good to talk to everyone. Hope to see you on the next one.